Right. Thank you. First of all, thanks to you, to everybody, for being bee supporters. This is really an important mission to try to help. It's one of the things we can do. There's lots of disease problems, pesticide problems, but underlying everything is food, nutrition. Same for humans, same for bees. So thanks for coming and thanks for supporting the bees. I'm going to start with the acknowledgements because I want to emphasize that this talk is, or this work, this research, we've started, well, it's 2009 that we started, we've been going for a while, is not a one-person job. And we have partners, GNS, Landcare Research, Assure Quality, Eastwood Hill, Foundation for Arable Research, Federated Farmers, National Beekeepers, etc. They are giving us their time, and they're our partners in doing the science work. And John Hartnell, Barry Foster, John McLean, and Paul Badger have helped. We really need the help, and they've just done a wonderful job in Gisborne these last 2 years. Our platinum sponsors, we especially want to thank, especially Inglebee, one of our best sponsors, who's not only helping us financially, but helping us with a huge demonstration farm, which you'll see tomorrow. Foundation for Arable Research in New Zealand, Manuka, Midlands Apiaries, Airborne, Kintail, and Hawke's Bay. And then we have many, many other sponsors. So I want you to understand everything I say today is based on all this help. I'm going to talk about three things. The bee plant diversity in general. We know from what Maureen has said and everybody else has said that diversity is key. Just like humans, you can't just eat one substance and survive well health-wise. Then I want to talk about the specifics of the flower, my favorite topic, which is the pollen quantity and quality. And then finally, the hard part, which is the plant quality and the suitability of the plant for the farms. So for bee plant diversity, we started in 2009, and those bee plant guides, you can download them from the internet, from our website. They stand. They're very strong and good. The plants on there were the plants that the beekeepers selected. And they're very good guides, but we've gone beyond that. And we asked a very critical question. Okay, plant trees for bees, but why don't we maximize our results and plant the highest protein pollen for the bees? And that started our fieldwork. This is Sustainable Farming Fund. And in the first year, in 2011, in the first grant we got from Sustainable Farming Fund, we collected 50 species of plants from Canterbury and 50 species from Eastwood Hill. Here you see, it's already been pointed out to you, that's the pollen basket, and that's a big one. It's a good one. And that's what we're looking for. That's on a weeping beech tree here in Gisborne. So we're collecting those from the bees. The first year at Eastwood Hill, Jean-Noël Galliot, a French intern, came to help me. And I said to the curator at the time back then, I said, well, what do you think? You think we got it all? You know, we got 50 species. Well, there's 1,663 species here. Just the species, I'm not even talking about the cultivars at Eastwood Hill. So the curator said, well, I think you just scratched the surface. So when we got our second grant, I wrote into it that we're coming back, and we're coming back for a whole year to watch what goes on here. So we came back spring 2013, and thanks to our 3 student interns, we did well this year. They're going to kill me for showing this picture. It's in our little special file called Picnic, and no one's supposed to see it, but they were fun, and they worked hard. This, seriously, was the dream team. They got 100 new species for our study because they systematically, day after day after day, boots on, combed through Eastwood Hill off the path in a group of 3 of them. I was with them when I could be, which wasn't all the time because of admin duties, and they searched for something in flower. And I'll explain to you why we need something in flower. So what did they do? We had priorities. Number 1, first thing you want to do is capture some bees with some pollen loads. Number 2, the second thing you want to do is herb-bearing specimen. If you don't have those 2 things, I'll show you why later, it's not a good sample. Then we want to get flower samples with pollen, 2 different types of pollen. And then for gravy, we want the photographs. I'll tell you why later. So 100 new species, it was fantastic. They went to any lengths to get this data. I couldn't believe the things they did when they came back with some of their photos, and even when I was with them, they were persevering. They would not leave until they got the pollen, the bees, and the samples. This is them catching the bees. We had poles that went up to 4 meters, and this is me and Jean Noel. We are doing the herbarium specimens. That is very, very critical to our work. There we're drying the specimens, and here is collecting the actual pollen from the actual flower that we got the bee out of. And there's, again, he's making a frozen flower specimen, and he has to make sure, it's very meticulous work. We had a hand lens, and you had to look at the flower and make sure that the bees hadn't taken all the pollen away. It had to have pollen in it in order for us to make the slides after that. This is what we did with it. We used a little cube of jelly with Fuchsin stain, and we stained the slide. Now that makes one type of pollen sample to identify and verify that it's that pollen. And we put it on a slide, and those all go to GNS Science. And then the other thing we did, we collected more pollen, and that will go into acetylysis, which is a different procedure to look at the pollen in another way, for more scientific detail. Then we photographed the pollen. All 3 of the students were excellent at photography. I'm still not, because I've always had the students do it, so I'm still training. But they were very good. We used a Canon macro lens, Canon system with a twin flash, which gives you very good 3D close-ups. Did you see the little bee there? So this is a good photography. We were told you can't do this, but with practice you can, and with a twin flash. And then we take a picture of the whole tree and the branches, etc. And then we come home and they put all the herbarium specimens in, do the jelly slides, and put the photos into the database, a lot of database work. So why did we do those collections? I just want to explain to you that the herbarium sample, if I get a collection of pollen, I don't have a herbarium sample, I can't use it. The herbarium sample verifies, because I'm a botanist, you have to have a sample. It has to be checked by a more knowledgeable botanist than myself, so that you know that that species that you got that bee out of is correctly identified. The bee pollen loads is for protein analysis, and we take a little bit of that little amount of pollen that we give to the lab for protein analysis. We take a little bit of that out and make a microscope slide. That microscope slide of that pollen from the bee pellet has to match the sample references, the acetylized and the jelly slide one. If it doesn't match, then we've got it wrong, and that happens. We've had that happen once where a bee was in a flowering ash tree, but it was actually had a pollen basket full of hawthorn, which was the same color, white. And these pollen samples are for reference. We're doing the Fuchsin stain primarily for the beekeepers and the honeypackers, because you don't always do acetylysis. It's very expensive. It's a lot of scientific detail, and sometimes you just need a jelly slide, you know. And so we're doing both all the time. The flower samples are for me, so I can play with my flowers in the freezer after. And also I've got some things that I need to do for quantifying how much pollen there is per flower, which I'll talk about later. And the photographs. We're building our photograph collection for future catalogs. Catalog of the plants, the pollen, and the pollen pellets. So we got 100 species in 2011, 2012, and 100 species in 2013. And when we hit 200, we had champagne. It was a great night that night, and because one of our French students was from Champagne. So the new total number of species for which we have already 100 done, and we're in the process of doing the next 100, we've got 200 samples of plants, which we know the protein. Now, we combine this data that we get in the field, all those 200 plants from the field work, with our data from the literature search. And I'm also doing, that's what I'm doing when the students are having fun in the field, I'm doing the database and other things like that. And we've got for New Zealand 345 species, and we now have from an international literature, which was very important because if they say that it's a good bee plant in Britain and we have this plant here in New Zealand, it'll be a good bee plant here probably. And if it's not, we need to know why. And our collection in our database is now about over 800, and I'm only about a third of the way or a half of the way through the international literature. So the overlap between these, we have to discern which ones of those international plants are also in New Zealand, and are they okay to have them in New Zealand in terms of planting them out. And then we have protein data for some of them, but it's interesting. I was quite shocked to find that a lot of the things we have here at Eastwood Hill that we see is very attractive to the bees. We've got protein samples. It's not on the New Zealand plant list, bee plant list, and it's not even on the international. A lot of that is, as Ben showed you, a lot of the plants here come from China, and those bee plant lists were made up in Europe and North America. So there's resources here at Eastwood Hill that New Zealand can capitalize on, and because of the HSNO grant, these species are already here in the country. We just have to make sure they're not going to be invasive. Native plant species, that's very, very, very well covered because of Butz Hearn's article in 1995, very well covered in the literature of what bees go to in the native plants. But darn hard to get the protein. I can't tell you how many days I've spent in native bush trying to get a sample. So we got, I'm just going to rip through them right now. We got Phormium, 32%, great pollen. We got cabbage tree, great pollen. Anything above 25% is high. Terata, Pittosporum genuides, great pollen protein. Five-finger, critical winter resource. Good pollen, 20%, that's still good. But we had to get Kanika and Manuka pollen, so we went up to the East Cape. And at the time, there was a bit of a question I was being asked by many people, do honeybees take pollen? Does pollen get into the nectar? At that time, people were asking me those questions, so I thought, well, okay, we'll look at that question, and I'm going to get the protein. But this is me and Jules. The honeybee that we looked at in Tiki Tiki and Te Aroa and at Ruatahuna, they were not taking pollen from Manuka. They're very tiny anthers, and there's better stuff out there. The bees were on the flax and on the pasture herbs. They were not bothering to take pollen from Manuka. But the native bees were focused on it. It was fantastic. The native bees, some of them come in like dive bombers and rip through, and some of them come in and they spend ages, and there are many, many different native bees, and they're all in the Manuka taking pollen. They also take nectar. The honeybee primarily just takes the nectar. I'm not saying it will never take pollen. I've heard beekeepers tell me it can and does. In certain circumstances, it would be when there's nothing better around. And there we have, yes, the answer to the question, pollen does get into the nectary because it falls off. If it's not falling off a honeybee leg, it will be falling off the native bees. So those little yellow spots are the pollen. So in essence, we've got over 800 candidate species that we're playing with. And we've got protein values, or we'll have, when this year's over, 200 species with protein. And we're building up the content in our photography and all our literature search for these catalogs that we want to do in the future someday. It's not part of this grant, but it's hopeful for the future. Now I want to consider, okay, what makes a good plant? When someone asks, oh, okay, you give us a list of 800 or 300, well, which ones are the best? This field of, someone asked this question today, field of bee nutrition is an emerging field. We're now starting to find, and I'm very happy because Doug Somerville did his work around 2000, we're now starting to find that a lot of new papers are coming out, and thanks to Maureen for that one from Pasquale. They're starting, and the point is, if we can do for sheep, if a farmer can tell me for a sheep, well, this bunch of grass gives us so many, what is it, metabolizable units of something, and it grows this many sheep and thing now. Well, we can do that for bees. Why not? But in order to do that, we need to know a little bit of information. The protein results, you can download this list from our website. I've selected some. We've got more than 59. We've got 100 done, but I selected 59 that we could put on the website, and I showed you whether it had a high, medium, or low protein in it. Protein's not the whole picture. There's other issues to consider, like fat and amino acid balance and stuff like that, but we're just doing crowd protein as a first step. And so what makes a buzz plant? When we're doing our research, sometimes we find the bees because we hear the buzz. And I'm sure most of you know about that. And if you hear a loud buzz, you go, wow. And you find the tree, and there'll be hundreds and hundreds and hundreds of bees on it. Now, theoretically, and I do say theoretically because I don't know anyone who would ever fund anyone to do this meticulous work, you could weigh the amount of pollen in an anther, multiply by the number of anthers in the flowers, the number of flowers in the inflorescence. Inflorescence is just a cluster of flowers, and how many clusters of flowers on the branch and how many branches on the tree. Now, in my imagination, I do this exercise all the time because when I look at a plant, I go, okay, a bee needs 120 to 145 milligrams of pollen to be able to get from egg to adult. So if that anther had that much weight in it, how many, you look at an oak tree and you think, wow, is that like worth 30,000 bees? It could be. I just have to, one day we'll get get this going. No, but then there's other things that are really important in what makes a good bee buzz plant. The ease of access. Is it how much work the bees are like us, cost-benefit analysis and everything you do, how much work is it to get this? And you got dish, bowl, brushes, catkins, they're easy, they're open access, no problem to get to that pollen. But directed access flowers, there's lots of tubular flowers. I went to one farm up in the high country and they thought they had good bee plants. And I said, well, that's great for bumblebees, but those flower tubes are too long. A bee tongue is only 0.7 millimeters. And that's a 13 millimeter floral tube. And if the nectar's at the bottom of it, they can't get it. They might be able to get the pollen at the top, but not the nectar. So the floral tube is really important. And then they have these closed access. A lot of people think these are nice, but I don't really like these flowers for bees. I don't think sometimes honeybees don't go to them, like in Robinia, pseudoacacia. We just don't see the honeybees. They take the nectar, but they don't take the pollen. And then I open the flower. It's hard to get that pollen. A lot of work. Bumblebee can do it, but honeybee can't. And then landing platforms. The bees got to be able, bees don't hover when they take pollen and take nectar. They like to land and stand on something. So I'm just going to show you some flowers. And this is a Super Bowl flower, camellia, fantastic winter source of pollen, and a very, very good bee plant. This is magnolia. They don't go to magnolias that much. I don't know why. But also excellent bee plant. Tons of pollen per flower, but not many flowers per tree. Apple has an open flower. You can see that the bee is hanging onto the petals there. You just look and see in the photos where what the bees are hanging on to. So then you multiply it up. There's a cluster of flowers and inflorescence. And there's the tree. That's lots of pollen. And there's an ornamental apple, same thing. This bee here, what it's doing is they scrabble around and collect up the pollen, and then they pack it in their legs. They leave the flower, fly back, and then pack their legs, and then they go back in. This is on certain kinds of plants. Photinia biverdiana, unbelievable source. Ceanothus, that's even more aggregated, the flowers. So that's easy for the bee to just climb around and get stuff. There's Acer. This is our October answer. Maples. Flowering nectar pollen in October during that pollen crash. And there's the Japanese maple. There's a melaleuca, that's a brush flower. Quercus, that's catkin. But when you look at a quercus, there's the little person down there in the little red circle. That's me. And that's the one that I did an imaginary calculation of that 30,000 bees there. It grows for 100 years. There's another one. There's our beech. And they're huge. We're going to see that on the lunch hour tour. This is a flower. This is a tubular flower. And we see the bumblebees in it, but we don't see honeybees in it very often. But there is a honeybee up there in the flower. But that's rare. And this is the closed flower. This is your bean flower, and it's not that much fun for the bees unless it's the right size. If the bee is, in relation to it, strong enough to open the flower, no problem, but otherwise, leave it to the bumblebees. And you can see there, the honeybees, they're trying to struggle, and then they end up robbing. They go to the base of the flower, and they end up robbing if they can. This is a bee heaven, and there's 3 bees in there. If you can see it, it's a test. There they are. That's bee heaven. I couldn't believe it when I saw that, going through the photos. I thought, oh my gosh, what is that? What is it? We can plant it on farms. Well, what plant is it? Huge amount of flowers in there. Good guess. It's a palm. It's a palm. Yeah, it's like a cabbage tree. Yeah. Well, I don't know if you can grow that on a farm. We'll ask Angus. I don't know. But yeah, well, yeah, I know. Everybody always disqualifies all my plants. Okay, so can't do it. Okay. But anyway, in principle, we've got many levels from anther tree to quantify the amount of pollen. That's what we're trying to do. Even we'll do it qualitatively. I don't think we can do it quantitatively. And it depends on ease of access and landing points. There's other things that John has mentioned, like the smell of the pollen, the attractant, the cues, and scent and stuff like that are important. And the cost-benefit ratio, always think about how hard is it for the bee to get that information. So now the last part of my talk, plant quality and suitability. Now I'm in trouble because I am not a landscaper, a forester, a farmer, a beekeeper. I'm a botanist, and I can do the flowers, but when it comes to plant quality and suitability, we've got these lists. I know that 2009 list we did of 98 plants, that stands because we worked with the beekeepers and the nursery people and the farmers to work through that list. They were ranked by beekeepers, etc. So, but each of those bee plant guides that you can still download, and it's very, very valuable. They stand. There's nothing wrong with them. It's just that we're doing more. We had 25 native species and only 16 exotic, which was one of the reasons why I wanted to come to Eastwood Hill when Barry Foster told me it was a good idea to come here. We need more exotics on our list. But those stand, that's fine. And we're adding species all the time, and we have to go through those species. We've got to evaluate each plant on a number of factors. Is it invasive? You just told me. I just lost my trichanther. I mean, you know, I keep losing plants that I think are good bee plants because of invasiveness, or it's toxic, like horse chestnut turned out to be toxic. Now I find out bad honey. I just read some literature that said from way back in the 1916 or so, that we don't want flax around our clover because our clover honey gets tainted with flax honey. I'm like, well, okay. And then other nuisances, a farmer say to me, I don't want to have those branches falling down like cabbage trees, etc. And the expense, we can't plant like Grevillea and Banksia integrifolia are really good plants, but very expensive. So, etc., etc. And the flowering calendar is what we have to produce. There is one you can download. It's based on what we have data for, which is 163 species. They're at the national level. We do not yet have flowering times at the local level because it'll start earlier in the north and later in the south, and their durations will be different depending on where you are. So that's all work in progress. And there's the availability of how many plants we have and when they flower, how many species. It's a simple sort of flowering chart. The Trees for Bees demonstration farms. Here's our progress report. I'm proud to announce, thanks to a lot of people doing a lot of work, including the farmers and beekeepers, we have now planted 9,500 trees since 2011. And we've covered 8.1 hectares of land. And we've completed 5 farms with small plantations. We are partially planted 5 farms in that they're in the works. Some plants are in, but we're still got more to do. And we've assessed greater than 5 farms, and I actually think it's more than that already. And the farmers and the beakers have been fantastic in helping us. And the farms represent arable, sheep and beef, livestock and orchard, queen raising, apiary. Now we're getting into more dairy, high country, etc. We're trying to get representative diversity in our farms where we plant our trees. And it's an iterative process. So I get the results from the database and the literature and the field work. And then we talk to the advice from farm planting advisor, farmer and beekeeper. and it goes round and round in a circles. And I just want to introduce Angus, our next speaker. Angus McPherson has joined our team. Thank heavens for us, because he does know how to plant, practical experience as a farm planting advisor. We are together developing principles and guidelines to how people can customize our work later on for their own farms. Each farm has a different set of requirements and planting opportunities, and at the end of Angus's talk, there'll be a brochure we hand out, which is a summary of how to plant on farms. Thank you very much.